Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 22nd September 2020. And today, for a change, we are going to talk about the first experiment that you need to perform once you come back, and the background is this. So as I have been telling you since 1 year, when you are in 11th class, there are some major headings whenever you are performing an experiment that are to be written. First one is objective or aim. Second one is the required materials. And third one is procedure. And the fourth one is observations. And the fifth one is precautions. And of course, after that, you need to write a result. So after observations, based on observations, you write result or conclusion, then you make precautions last. And of course, on white sheet, you need to draw the diagram. So this is what you have to do in your biology record. So your lab assistant has helped you to see how exactly in reality the pollen germination takes place. So for that, what is required? So we start off with aim or objective. What is the aim? To study pollen germination on a slide. What are the materials required? Fresh seasonal flowers. Of course, we have picked up hibiscus and clean neat slide, cover slip. Of course, we have got compound microscope, sucrose, boric acid, magnesium sulfate and potassium nitrate and the beaker. So procedure, before we actually do the experiment, we need to use all these chemicals to prepare a nutrient medium. So how to do that? So prepare a nutrient solution by dissolving 10 grams of glucose, 10 grams of boric acid, 30 milligrams, see mg, 30 milligrams of magnesium sulfate and 20 milligrams of potassium nitrate in 100 ml of water. So what is that you have to do? You take a beaker, in that 100 ml water you take and add all these chemical substances as mentioned in quantity, like 10 grams, 30 milligrams, 20 milligrams, like this. And after that, take a few drops of this solution on a clean slide and dust few pollen grains from the stamen of mature flower on it. We have used hibiscus, so we have dusted that. Observe the slide in the microscope after 5 minutes and then observe it regularly for about half an hour continuously with breaks. So compulsorily you must wait for 10 minutes at least. Now what is that you will observe? In nutrient medium, the pollen grain germinates. The tube enlarges and comes out of the pollen grain through one of the germ pores to form a pollen tube. As you are seeing on a pollen grain, there are so many germ pores surrounding it, and through one of the germ pores, pollen tube starts coming out. So the tube nucleus descends the tip of the pollen tube. So there is a tube nucleus that's coming out, and this, the generative cell, as you may recollect from your theory knowledge yesterday, the pollen grain will have two cells, isn't it? One is the generative cell, another is vegetative cell. So from the generative cell, it passes into it, soon it divides into two male gametes. So tube nucleus will undergo mitosis, divides and forms two male gametes, male gamete 1, male gamete 2. If you recollect again from your memory, this is linked to double fertilization. So male gamete 1 will combine with ovum, functional ovum, remember megasporogenesis, and forms zygote, and that is called syngamy, fertilization 1. And male gamete 2 combines with the two nuclei, polar nuclei, it becomes a triple fusion and forms endosperm. We said pen, primary endosperm nucleus. So that is fertilization 2. So these two male gametes will involve themselves in both the fertilizations. Fertilization 1 Syngamy, fertilization 2 triple fusion. So Syngamy plus triple fusion is equal to double fertilization. So this is what the purpose of the formation of these two male gametes. Each male gamete is either it is spherical in outline, or sometimes it is a little bit disc-shaped, as you observe under the microscope. And of course, the entire pollen grain will have two layers, exine and intine. And what is the result? What is your purpose? To study pollen germination on a slide. So once you achieve your purpose or aim, that should be your result. So what is that you will write? The pollen germination under the compound microscope is observed and noted. That should be your result. And what are the precautions that you should take when you are performing this experiment? Flowers should be fresh. You should not take old and rotten ones. So fresh flowers should be used. And then the slide that you are using, wherein you are dusting the pollen grains, should be neat and clean. Otherwise, if it is dusty, instead of pollen grains, you will see dust particles. Hence, take care of that. And third, as usual, no air bubbles should be there. If you allow air bubbles, then you will see not pollen grains but air bubbles. So take care of these three. Now, what are the precautions that you need to take care while writing, while performing the experiment? These are the three precautions you should take. What are the precautions that you need to take when you are writing the experiment in the record? And since you are doing the experiment with your own hands, I want you to write procedure in the past tense and point wise. Like here, prepare a nutrient solution is there. So what you will do, a nutrient solution having decomposition is prepared. That means is prepared by me. That's what. And second, take a few drops of this. Don't write take a few drops. Few drops of this nutrient solution is taken on a clean slide, like that. I hope you understood. So procedure should be past tense and point wise. Similarly, observations also should be in the past tense, because you have already observed. What you have observed, you are writing. So the observation should be again in the past tense and point wise. And then result, of course, you can write. You have seen whatever result you get, you write. And then precautions, of course, you can write in the present tense. So objective, present tense, requirements, present tense, procedure, past tense, point wise, observations, past tense, point wise, precautions, present tense, result, past tense. So this is how you need to do it. And also this is how the dusting of the flower will be done. Pollen grains on a clean slide. And then you can see how exactly we got yesterday in the lab. This is how the pollen slide has been prepared, and germination just started as you are observing. And then finally each pollen grain started giving pollen tube, germination of pollen tube. So this is what you need to do when you come back to the school. And before that, you need to have a record, maintain a separate biology record and write down. And this is to be submitted to CBSE. Hence, you need to write with a lot of care. Draw beautiful diagrams. Write neatly. So this is experiment number 1 related to second chapter, sexual reproduction in flowering plants. In my next session, I will come again with one theory topic and then related practicals one. Until then, keep writing, maintain your record, and draw diagrams, post in our WhatsApp group. Take care.